Good morning. Hello, my AP biologists. So, this is your Chapter 5 video lecture. I've decided to break it into two chunks. So in this video, we're going to go over carbohydrates and lipids. And then in the next video, we'll cover the two other macromolecules, which are protein and nucleic acids. Okay. So I would like to just quick reminder, our four important organic molecules. We have carbohydrates, lipids, proteins, and nucleic acids. And we'll be focusing on 1 and 2 in this video. I did get some requests to just do a quick reminder about dehydration, synthesis, and hydrolysis. Okay, so I'm going to just do a quick review over that. So I'd like you to think of these macromolecules, which we call polymers. Poly means many. And the reason that we have this word polymer in there is because they are made up of many bonded monomers. So just like a brick wall isn't made of just one single brick, you have to put many bricks together to build this brick wall. I want you to think of monomers and polymers similarly. So one monomer is the brick, right? And the polymer is the brick wall. Okay, and so a quick reminder of what the bricks are for each of our polymers, okay, each of our macromolecules. So to build a polysaccharide, which is a carbohydrate, that's the polymer, our monomer will be sugars, and we're going to break down even more detail what those sugars are. Okay, to build our macromolecule, which is fats, our brick is fatty acids. To build our polymer of a protein, our bricks or monomers are amino acids. And then to build nucleic acids like DNA and RNA, our bricks or monomers are nucleotides. Okay. So how do we put these monomers together? So if I want to bond two monomers together, I need to undergo something called dehydration synthesis. And so what exactly is happening in dehydration synthesis? Go ahead and watch this little animation. But hopefully what you can see is that you lose an OH from one of the carbohydrate sugars, one of the sugars, and you lose an H group from another one. And so we know 2 H's, 1 O is H2O. So by removing one molecule of water, we are able to now bond these two monomers together. And you can see here they are bonding at the O group. Okay, now what if we want to break down these polymers? Meaning they've been bonded and now we're trying to break them apart. So let's break the word down. Hydrolysis, hydro means water, lysis means to cut. So hydrolysis literally means to cut by water. And so you can see here, this kind of shows the full scope of animation of first dehydration synthesis, which is right here, right? And now we have this water molecule that is going to break this bond between the two monomers. So let's watch it break the bond, and those the OH group is reattached to the one monomer, and the H group goes back to the other monomer. And so that water molecule's atoms are placed back into each of the respective monomers. Okay, so now it went from polymer to monomer. Here we go. Let's see. Right now it's a polymer. We add water coming on through to break that bond. It's moving up, and now we have two monomers again. All right. So now let's talk about our first big macromolecule, which is carbohydrates. Okay, and so what is the brick for carbohydrates? Hopefully you were thinking it is glucose. Okay, and so when we look at a chemical formula for glucose, we can count the carbons. Okay, hopefully you remember that glucose, the molecular formula is C6H12O6. And if you were to count all of the atoms in this glucose, that would confirm it. Okay, but what we can do here is we can number the carbons. And so just so that you know, carbon 1 is right here. Okay, and then we go around carbon 2, 3, 4, 5. And then carbon 6 is always going to be kind of popping up like a little chimney. Okay. And so again, if we count all of our, and then keep in mind in this corner, you'll always know this is going to be an oxygen. Okay. So now if you want to break a sugar down, just to also give you another reminder here, if you want to break a sugar down, it ends up being that each component is CH2O. And that probably sounds familiar from when we were taking biology together the first round, where we remembered CHO, CHON, and CHOMP. So for the carbohydrates, remember that your the parts of the glucose molecule, you're just going to keep seeing CHO, okay? So now let's move into a way that you can write this to make this a little bit simpler. So let me get my head out of the way here. So, and maybe also make myself a little bit smaller. So it's not working for me. I guess that's the smallest I can get. Okay, so to simplify this, we don't have to write our carbons. We can assume that they're there, right? So this is the molecular form here, where we see the carbons, except for our oxygen up in this corner. But you might see this more simplified form where you don't see the carbons, except for our carbon 6, the little chimney. But we assume that these are carbons, okay? And then you also could very likely just see it like this, where there are no carbons present. You don't see even the chimney. And you just assume, though, when you see this shape, that this is a representation of glucose, the monomer or the brick for carbohydrates. Okay. So let's see. I want to just quickly show you what we're going to have. We're going to put this into a chart in just a moment so that we can recognize this shape. Okay. So we already learned about dehydration synthesis, but let's just show it for our monosaccharides or our monomers of carbohydrates. So what you see here is the OH group on one of the monomers and the H group on another one of the monomers. We lose those. We dehydrate them, right? We take one water molecule away, and in doing so, it undergoes a dehydration reaction. And as you can see, we now have these two glucose bonded together, okay, and bound at the oxygen right here in the middle, kind of like they're holding hands, okay? And the output then is this water molecule and this disaccharide, di means two, this disaccharide called maltose, okay? And we can also, we just talked about hydrolysis at the beginning of the video. This maltose disaccharide, if we were to break this right here, we would break it with a molecule of water and it would return back to a glucose plus a glucose, those two monomers, okay? So, let me get out of the way again. So these are the important disaccharides that I want you to remember, okay? So if you look at your notes, and let me just make sure I'm with you here. So if you look at your notes, for monosaccharides, it says quick energy monosaccharides. Next to it, write like glucose, and they contain 6 carbons, okay? Monosaccharides, they're quick energy. They mostly contain 6 carbons. The exception is fructose, which I also have there for in your notes. That has 5 carbons, okay? For the disaccharides, also considered quick energy. And now we're just going to, I've given you the 3 important ones I want you to remember, but now let's break down what those components are, okay? So a lactose disaccharide, which we see right here, That is it's milk sugar. So it's the sugar that we find in milk. And that is a glucose plus a galactose. Okay. And so you can see here, hopefully you recognize that this is your glucose. Okay. And then we have our galactose. Okay. We bond them here. Just like before, they're holding hands at the oxygen after they've undergone dehydration synthesis. Okay. What is maltose? So maltose is two glucose bonded together. So two monosaccharides of glucose bonded together. And you know what? Let's rewind for just a moment here. Rewind here. Okay, so quickly in lactose here, I misidentified for you. This guy right here is our glucose. Okay, and this here is our lactose. And hopefully we understand that all of these monomers, they are all isomers of glucose. Okay, so they all share that same chemical formula, except for the exception is fructose, because it only has 5 carbons. Okay, so we were at maltose. Those are two glucose bonded together, disaccharide. And then finally, we have sucrose, which is table sugar, and that is a glucose bonded with a fructose. Fructose is the sugar that we find in fruit. And so when we bond these two monosaccharides through dehydration synthesis, so they lost a molecule of water, they're joined here at the oxygen, like they're holding hands, and we have sucrose. So in your notes, sucrose is table sugar, glucose plus fructose. Okay, and just to also clue you in terms of lactose intolerance, if you have a question about it. So there's kind of two big reasons that someone would have a lactose intolerance. The first one would be that you're either allergic to lactose. So your immune system sort of triggers this immune response and an allergic reaction to get rid of the thing that is causing your body to have a reaction. The other big reason, though, is that a lot of people don't have the enzyme that allows you to digest lactose. Enzymes are proteins. We learned a little bit about enzymes in the last video, but enzymes, they help catalyze reactions, including things like breaking down and digesting. And so if you don't have the enzyme to break down lactose, and that enzyme is called lactase, okay? If you don't have that enzyme, then it can cause some serious indigestion, okay? So on your notes, make sure that you have filled in those important disaccharides. All right. So now we're moving on to polysaccharides. So something that all polysaccharides have in common is that they do not dissolve in water. They're not soluble in water, and they are too large to pass through cell membranes. Okay, they need to get assistance. They are so large that there's no way for them to get through cell membranes. Okay. So there's two big categories of polysaccharides. They are either used for and all about energy storage, or they are going to be building material, structural material to build cellular structures. Okay, so let's start with starch. Okay, so starch is how plants store sugar. Okay, and so you see these repeating units. Okay, and you see these branches off of more of these units of bonded together monosaccharides. Okay, that's just because we're trying to pack in as much energy storage as we can into this space, in particular within the cells of our plant cells. Okay, so I want you to go into your notes. Want to make sure I'm getting you for polysaccharides. Okay, so polysaccharides in your notes, not soluble in water, too large to pass through cell membranes. Okay, and in terms of their energy storage, starches are the energy storage found in plants. Okay, so and also let's remember, what is the process to make those sugars in the first place? Did you say photosynthesis? Yes. Okay. So that's how they make all their sugars. And then they can store any of those excess sugars as this polymer called starch. And they store that within their plant cells. So our bodies and animals in general, we do something very similar. We don't store starch for energy. We store something called glycogen. So our bodies will store sugars as glycogen. So when we eat food that is built out of sugars and you digest it into simple sugars, okay, it will then be reconstructed as a storage form. And it'll be stored as this molecule called glycogen. And it's stored and put in your liver. Okay. So when you eat a bunch of carbohydrates, your body breaks those down into more simple sugar, and then they reconvert them and construct them into this form called glycogen. And when your liver is full of glycogen, what ends up happening is that any of the excess sugar that you then take in will now be converted right into fat, which is also a really important macromolecule. Okay, so for our notes, energy storage, part B, glycogen is found in animals. Okay, so when you are using a lot of energy for maybe going on a run, okay, your body is using those glycogen stores to get the energy supply to help you do those things, okay? And again, you can see these branched off polymers from the polysaccharide. Again, the whole purpose of that is to pack in as many of those bonded monosaccharides as possible. We're trying to get as much energy as we can to store. Okay, so outside of energy storage, we also have a huge use for structural components. Okay, so polysaccharides are also critical for being structural molecules. So let's first talk about cellulose. Okay, so plants let's refer back to our starch that we were just talking about, okay? So plants can use those sugar stores, like their starches, to help build their cell walls, okay? So as they're growing, they need to build more cell walls to continue to build more cells. And so these cell walls are constructed out of a polysaccharide called cellulose, okay? And cellulose, we know, is indigestible by humans. We can't break it down, so we have to use our molars to break down those plant cell walls and be able to capture the nutrients that are within the plant cells. Okay, so in your notes, it says that cellulose builds plant cell walls, and this is really critical. It is the most abundant molecule on Earth. Most abundant molecule on Earth. That is cellulose. Okay? So let's now move on to chitin. So chitins are another really important structural component. They are also polysaccharides, okay? And chitins, they form things like exoskeletons, like we can see here with these arthropods, and they also build fungal cell walls. So remember, a fungus, like mushrooms, those are not plants, they're fungus. And so chitin, instead of cellulose, it's chitin that builds their cell walls. Okay, and I believe that that is already in your notes. Fantastic. So now it's time to move. Oh, let's actually first address our summary table. Okay, so this is a really good opportunity for you in your notes to. I would do this by hand, right? And I will be sharing out in this presentation and in the next video as well. You know, I will do the summary chart for you, but I also really recommend that you try to either test yourself by creating one on your own or trying to create it on your own first and then referring back to mine. Okay. So if we quickly summarize our information from carbohydrates, okay, what is the monomer and also what is the structural shape so that I can identify it on a test. So The monomer are sugars or monosaccharides, specifically glucose, fructose, and galactose. Okay, those are the important monosaccharides. And then we have our disaccharides, maltose, sucrose, and lactose. What is bonded with what? Maltose is glucose plus glucose, sucrose, glucose plus fructose, lactose is glucose plus galactose. Okay, and then what are some of their functions and the polymers? So one structural polymer or polysaccharide is starch. We have glycogen in animals. We have structural polysaccharides, which are cellulose and chitin. Okay, so now it's time to move on to lipids, those fats and steroids. Okay, so two different categories of lipids. We have fats, we have steroids. We're going to go through both. All right. Besides fats and steroids, there are some other types of lipids. Okay, we have oils, phospholipids, and waxes. Okay, and so the basic lipid structure is that there's one glycerol molecule bonded with 3 fatty acids, and we're going to go through that. Okay, and so it'll be one glycerol, and those 3 fatty acids are bonded by a carbon, carbon, and a carbon. Okay, and so you can see. Let me kind of go back here. I don't have a picture of those up yet. Just to quickly remind you about what a phospholipid is. Okay, a phospholipid is it's a modification of a fat, okay? And so it's very, very important in cell membranes. We're going to be talking about it in more detail when we get into structures of the cell, but it is a modified fat. All right, so now here are some pictures for you, okay? So basic lipid structure. There's 2 types of bricks or monomers to build our lipids. Glycerol, fatty acid chains. So when I was doing this for glycerol, this right here is our glycerol. And you can see how many carbons, 1, 2, 3 carbons. And we have our 3 fatty acids, 1, 2, 3. Okay, and so what you can see is that there is a functional group on the ends of these fatty acids that look like an alcohol, okay? And you can see these rectangles here, and you can also see the functional groups here, okay? And so these here that you're seeing, these fatty acid chains, these are not drawn to scale. So normally these would be really very long hydrocarbon chains, like 16 to 18 hydrocarbons. So these are not drawn to scale, but it's just so we can fit it on the page. Okay, so for us to bond these fatty acids to our glycerol, we need to undergo a dehydration reaction, dehydration synthesis. And so we see here they're not bonded together yet. They're going to undergo a dehydration reaction. You'll see they'll lose 1 molecule of water for each bond they make. So they'll lose the OH group on this guy and the H group on this guy, the OH group on this guy and the H group on this guy. And same for the third one. And so that's 3 molecules of water are dehydrated out of these 4 combined molecules so that we can bring them all together. And now you can see they're bonded at the glycerol. Okay. And so hopefully you are seeing, you're going to see that carboxylic that carboxyl functional group on each fatty acid, that's where we're going to lose the OH component of our water molecule. Okay, so when you have this carboxyl group and then you have this long fatty acid, let's see, in your notes, I want to make sure that I'm not jumping. Okay, good. So in your notes for triglycerides, okay, it says a glycerol is 3 carbons, 1, 2, 3, and then a fatty acid chain So these guys, they're usually 16 to 18 carbons in a chain. So the fill-in is the word chain, okay? So now let's talk about the two different types of triglycerides, okay? So you have likely heard of saturated fats, okay, and hopefully also unsaturated fats. We talked about them in my class before. So first, I just want you to kind of observe these two and try to notice the differences, and then we're going to go through. Okay, so when I look at saturated fats, I see that the carbons are completely saturated in hydrogens. Okay, so there's no double bonds. It's all directly hydrogens bonded, fully saturated by these hydrogens. Okay, and so no double bonds. So these guys, because there are no double bonds and there's no kinkiness to their chain, they are solid at room temperature. Why? Because they kind of just stack right on top of each other, okay? Because there's no kinkiness in their chains. So they're able to just stack right on top of each other. So these are fats that are typically formed in animals, and they tend to be solid at room temperature versus comparing now to our unsaturated fats, okay? So unsaturated fats, we can see there's a double bond right here. So these carbons, they don't need to be saturated in hydrogen because their valence shell was fulfilled by double bonding to a carbon. Okay, these unsaturated fats are typically found in plants, and they are typically liquid at room temperature because of their kinky nature. Okay, and so if you think about, like, if you've ever cooked with canola oil, for example, it's liquid. When you're trying to stack these kinky fats on top of each other, they aren't going to stack nice and neat like the saturated fats, and therefore you see them liquid at room temperature. Okay, so in your notes, you need to fill in quite a bit for unsaturated. I gave you all the components for saturated. No double bonds, creates straight chains, solid at room temperature, usually from animals. For unsaturated, and I'll say it a little bit more slowly, double bonds, creates chains with kinks, liquid at room temperature, usually from plants. Okay. So here we have another picture, so you can see the unsaturated fat is very fluid, full of these kinky shapes here, versus our saturated fats, which are solid at room temperature, and you can see why they just stack nice and neatly on top of each other. Okay, so, all right, now I want to address something. Fats, they don't like water. They are hydrophobic. Okay, but we have solutions that do have fat mixed in. And so how does that happen? Well, what we have to utilize is something called an emulsifier. Okay, so let's kind of think of an example of, what do you mean a solution that has fat within it? So let's think about like ranch dressing. When you buy ranch dressing, it's not a whole bunch of fat and separated from the water. No, it is mixed together and it's this nice creamy substance. Well, the reason for that is because there's emulsifiers in it. Emulsifiers change the properties of fat, okay? So basically what happens is you add some polar groups, okay? You add some polar groups, and by having a polar end on your fat, it allows it to now dissolve into your ranch dressing. Okay, now something that's more important than your ranch dressing is like your blood plasma. Okay, so you have fats that are dissolved in your blood, and that's because they also have polar ends. Okay, and it allows them to move through your capillaries and also not separate out like the way that Italian dressing does. Okay, we also can use emulsifiers to clean fats. If you've ever, used like Dawn dish soap, it breaks down the oils because it has emulsifiers in it. Okay. And then finally, let's talk about bile. Okay. So you have these things, these emulsifiers are also in your liver. And This emulsifier is called bile, and it breaks down fats. Okay, where do you store this bile? You store it in your gallbladder. And so there are people who have to have their gallbladder removed. People who have their gallbladder removed, they have a really hard time eating fatty foods because they don't have the substance to break down those fats, okay? And then therefore easily digest the fats and then reabsorb it. So they should stay away from high fat foods, okay? All right, so now we have a picture here of a phospholipid. Okay, phospholipids are huge when we talk about homeostasis and cell membranes. Okay, so let's find the fat in the phospholipid. Okay, so we see our two fatty acid chains. Where's the third guy? We're about to talk about it. Okay, and we see, oops, Okay, we see our glycerol here, and instead of that third fatty acid, okay, instead, oh, and we can see it has that kind of kink leg here, so it's kind of kicking its leg out. So where is that third fatty acid? Well, it is replaced by a phosphate group up here, okay? And so this phosphorus, as you can see, it's surrounded by oxygens. And we know oxygens are very, very electronegative. Okay. And so they're going to, these bonds that are going to be very, very polar. And so it makes this whole head a polar head. Okay. And we know polar molecules, they like water. Okay. They're hydrophilic. Okay. But these long fatty acid chains, they are hydrophobic. Okay, so what we would call this type of phospholipid is something that's amphipathic, which means that basically it has two different properties on either end. It has a hydrophilic side and a hydrophobic side. Okay, and so let's go to your phospholipids in your notes. Okay, it says the third fatty acid chain, hydrophobic, is replaced with a charged phosphate group, hydrophilic, and it is the main component of cell membranes. Okay, so let's see. We've got that into your notes. Good, good, good. All right, so now let's talk about another category of fats called steroids, okay? So there are two main ones here. We see them, testosterone and estrogen, okay? And for these two main steroids, they are built out of rings of carbon, okay? And so you can see, hopefully, you're seeing the difference between a steroid versus, a testosterone versus the estrogen, okay? Which is, my thing is jumping around on me. Okay. When we see these steroids, we can immediately identify them because of their 4 carbon rings. 1, 2, 3, 4. 1, 2, 3, 4. Okay. The main difference that we see between these testosterone and our estrogens is that our, if we notice we have the same CH3 groups here. But in testosterone, we have this extra oxygen, double bonded oxygen that you don't have in estrogen, right? In estrogen, you have this OH group, okay? And then these double bonds, okay, those just mean that these blue lines are double bonds of carbon, okay? So in your notes, what are steroids? They are made of 4 fused rings of carbon. 1, 2, 3, 4, 1, 2, 3, 4, okay? Hopefully also these CH3s are jumping out at you as a functional group. Which one? Hopefully you said methyl. Okay. All right. So let's talk about, actually, let me just show you sort of the demonstration here. We can see, wait a second, where am I? Okay, yes. So we can see that even that slight difference in the component or the functional groups of testosterone versus estrogen, that it completely changes what is carried out, right? We have our male peacock, which utilizes testosterone. We have our female peacock, which utilizes estrogen, okay? Obviously causing huge differences, okay? And so the building block for these steroids is going to be cholesterol, okay? So this is cholesterol, and we are going to use that as our starting point in order to either build testosterone or estrogen, okay? So now let's talk about something called atherosclerosis, okay, which is having a buildup of fatty material in your blood vessels, okay? So this is someone who does not have atherosclerosis, no fatty streaks even, okay? But basically what happens is fat builds up in the walls of our arteries, okay, and deposits. And so what ends up happening is that it narrows the diameter of those arteries. And so unhealthy fat deposits in your blood vessels, they limit blood flow, okay? And so keep in mind, people think, and this also refers right back to the project that we did your sophomore year, if you did take me, that it's not just related to people eating excessive amounts of fat. If we relate it back to glycogen that we learned about in the carbohydrate section, right? If you are eating excessive amounts of carbohydrates, okay, and your body converts that into glycogen, stores it in your liver, if you go way beyond that and you keep eating way more hot Cheetos, way more cake, way more extra carbohydrates, what's your body going to do with all those excessive carbs after the glycogen stores are already all filled up? Well, they're going to convert them right on to fat. And so that will absolutely contribute to those fatty deposits. Okay. Last but not least, we have our waxes. Okay. And so waxes, you can think of like honeycomb, right? Or the shiny surfaces of leaves. You could think of your earwax. Okay. So basically they are long chain of alcohol. Okay. And so with one fatty acid chain. Okay. So one long chain alcohol with one long fatty acid. So here is your fatty acid and here is your long chain alcohol. Okay. And so you can see your carbon here at the end is double bonded to an oxygen. Okay. And that would give you, you'd have an OH. And then these two will join together and that will form a wax. Okay. So a wax, so let's just make sure that we kind of understand how those two are joining together. So because this is already drawn in polymer form. So you can see the oxygen that's holding hands, right? So on this guy, we lost an OH group. On this guy, we lost an H group, and hence why they are bonded. Remember, OH plus H, H2O. Okay, so what is the purpose of waxes? So in plants, it's a protective covering. It also helps slow water loss. It's an important part of plant structure, okay? And then for animals, it maintains our skin. It can trap dust and dirt, can help form honeycombs, especially like for earwax. That's good that it traps dust and dirt. We don't want that moving beyond and impacting, important structures in our ear. Okay, so in your notes, waxes are a long chain alcohol plus a fatty acid. They have a very high melting point, okay? High melting point. In plants, and then you need to fill that in, it's a protective covering. It slows water loss, okay? And then I already gave it to you what it is in animals, all right? And so also, as you are hopefully seeing through your notes, I have embedded a lot of images for you. I would encourage you to add more images if they will help you. Okay. So we are going to start off our next video with proteins. And now would be a good time for you to go back to your summary table and try to do the lipids one on your own before I give you the big reveal of the macromolecule table at the very end of video 2. All right.